The overall goal of this procedure is to isolate F1FO ATPase vesicles from rat brain for performing patch clamp recordings. This is accomplished by first removing the brain from a rat and homogenizing it. The second step is to disrupt synaptosomes by applying pressure. Next, the synaptosomes are layered onto the phycol gradients. The final step is to incubate with digitonin and lubrol to obtain the final vesicles for patch clamp recording.
An important function of mitochondria is ATP production by oxidative phosphorylation. The enzyme responsible for ATP production is the ATP synthase, located at the mitochondrial inner membrane. To study the ATP synthase function in detail, we isolate special submitochondrial vesicles that we call SMVs. They contain the ATP synthase enzyme in an inside-out configuration. We then record these SMVs with a patch clamp electrode, and we determine the leakiness of their membrane. That tells us about the efficiency of ATP production by the mitochondria. Demonstrating this procedure today will be Silvio Sacchetti, a postdoctoral fellow in my laboratory at Yale University.